And then you also have this one, the bacteria is fire blight. It's bacteria, and if you smell it, well, it's a plant pathogen, so it really doesn't matter if you breathe once, and it smells really bad. And this is bacteria. So for this one, I want you to do three-way streak. What is the purpose of doing three-way streak? Remember? What is the purpose?
Like different levels of pathogens. So like it's like most concentrated, and second most concentrated, and third most concentrated. You like spread it out.
Right. So that's how we do three-way streak. So basically, three-way streak is like a diluting process, and then at the very beginning, you get the highest concentration. So all the cells are growing together. You can't see colonies, but you see a pile of bacteria. But then at the end, when there are not so many bacterial cells, they separate, and then from each bacterial cell, a colony develops, so you can get single colony. There are some that are really good. My assistant did a very good job of getting single colonies, but some, the concentration of bacterial cells are so high that they're growing together. So three-way streak, the purpose is to get single colony, because after you get single colony, for example, one colony, all the bacterial cells have the same genetics, and then we can do sequencing, we can do inoculation. Whatever we do, we know what we're working with a single colony. So that's the purpose. And then how are we going to do that? You have inoculation loop in your box. So how are we going to do that is we are going to use a new plate. For example, this is the plate. This is the back of the plate. You can draw a line this way so that you separate the plate into three parts. And then the first streak zigzag line is going to be here, the first part. And then the second one, you drag some cells from the first zigzag, and then draw the second zigzag line. And then the third one is you draw zigzag, you drag some cells from the second zigzag and then draw the third line. So now I'm going to do a demonstration. So let's see. You need ethanol burner if you want to make sure everything is clean. And if you also have a beaker with ethanol in it, and then your inoculation loop, which is here. Before you burn the inoculation loop, make sure they're straight. Yeah, because later on when you burn it, you don't want to touch it anymore. So that's that. And then perfume your mother bacterial plate. And then I'm gonna get a new plate for demonstration, so I will just use this one. You can see that I draw the line here. So first of all, inoculation loop, I need to clean this. Actually I need to get culture prepared here. Okay, so I will burn the loop. The outer part of the flame has the highest temperature, so I use the outer part of the flame. So after I burn it red, then I put it in the ethanol. And then get some ethanol, dip in the ethanol, and then burn the ethanol. I'll keep the loop in around the flame because that's where it is clean. Microbes are everywhere, right? And now I'll open the mother plate And I'll move towards the flame as well. And then just sample some bacterial cells. And then I move this clean plate, open carefully around the flame, and then draw zigzag lines. After that, I burn the loop, get rid of the bacterial cells, and then dip in the ethanol, then burn the ethanol. So I need to cool down the loop. And how do I do that? I touch the lid or poke the media, the edge of the plate. And then after the loop cooled down, I draw the second zigzag line. Basically drag some cells from the first zigzag line and then draw the second one. That's it. And then burn the cells. Dip in the ethanol. Burn the ethanol. Cool it down by touching the lid. Then draw the third zigzag line. After that, burn the loop, burn the bacterial cells, then cool down in the ethanol. That's it. After that, I need to perfume my plate. So wrap the plate. And also wrap the mother plate as well. So that's it. And turn off the ethanol burner. So how can we tell if we get single colony or not? Well, after 2 days, the bacterial cells will grow. If you get single colony, you can see tiny dots of the single colonies. So if you want me to, apparently you're not going to come back after 2 days. So if you want me to take a picture of what you have done, you can have your name on the plate, label the plate really well, and then I'll take a picture and send you the picture, and you'll see whether you did 3-way streak successfully. So that's 3-way streak. And then the last thing is the nematode. So for nematode, I have set up everything, and you remember this is, what's the name of it, of this device? What's the name of the device? It's called Behrman funnel technique. So this is the most common way for nematode extraction. So basically, you have a funnel, plastic funnel or glass funnel, and then you have a wire mesh here to support 2 layers of tissue, Kleenex tissue, and wrapping some soil, and then have a flexible tubing connect to the funnel with clamp, close the tube. And the soil is actually from a vineyard. It's sandy soil. Usually sandy soil, by experience, I can get more nematode from sandy soil. And then get water, put water in it, submerge the soil in the water, and incubate for 24 to 48 hours. It has been incubated for 48 hours. And now I have the dishes, small dishes. You can put the dishes close to the tube. I can also help you with that if it's not, like if it's too high, then you can get water splashed everywhere. So just very closely, I can help you with that. And then carefully open the clamp and drop several drops of the solution, of the water, onto your dish. and then make observation with dissecting scope. So that's that. We have the device set up, and then because we have to share with another section. The section from 10 o'clock have already sampled several drops of water. So I am afraid that we don't see nematodes if we sample more because it's going to be diluted. So I just asked them to keep their nematode solution, and you can see that they're on your dissecting scope. So you can try to get more sample, but if you can't find a nematode, then you can always look at the dishes that are already on your dissecting scope. And after that, before you leave, I'm gonna ask you to toss everything here and then make a new one. So put new soil in wrapped with the tissue paper, and then put it here, and then continue doing incubation for my class for tomorrow. How's that? Great. I think that's that for demonstration, and you can get started. And while you do your work, I'll call your names, and you can start do observation of the samples in the middle bench. Questions? Okay, let's get started.